Good morning, professors and students. It is indeed my great honor and pleasure to share this with you, this particular topic on how gut microbiome impact on human cancer. Okay, let's first introduce the human microbiome. Actually, your body contains hundreds of different type of cells. Is the human body mainly made up of human cells? Okay, this is a particular question. The answer is not. Notably, the largest number of cells contained in our human body are not human cells. What else? Up to 90% of cells in human body are microorganisms. Your body actually consists of entire ecosystem of diverse and harmful little microorganisms, numbering over 100 trillion. So it's a huge number. What was in among these microorganisms? Okay, the human gut contains a collection of microbes that include commensal bacteria, as well as fungi, virus, archaea, and helminths. Bacteria make up 95% of the gut microbiome. Okay, and this picture is the human microbiome. It means that we each have a troop of good bacteria. We name it the bacteria superheroes to call our own. Thus, the greatest concentration of bacteria superheroes live in our gut. This ecosystem of microorganisms is a huge part of who you are. Did you know the function of your microbiome? Okay, they have many functions. I just give you some example. Your microbiome aids food digestion to fuel your body, influence your mood and energy level, helps you to adapt to your surroundings, to protect against harmful organisms, and much more. Scientists have only just scratched the surface of understanding how incredible these little guys truly are and how important they are to our health. Okay, it all started when you were born. Some of the bacteria in your mother's microbiome were passed along to you to become the beginning of your very own microbial fingerprint. When you were a baby, experienced the world for the first time, the bacteria and your microbiome were doing the same. By the time you were a toddler, the makeup of your gut microbiome was almost fully matured at 2 or 3 years old. Your bacterial superheroes had already established a natural and balanced state. Did you know that microbial diversity is a key to a healthy microbiome? Scientists have discovered that your microbiome fingerprint actually need to consist of many different types of bacteria to keep you strong, healthy, and robust to drastic or harmful changes. Did you know what a healthy microbiome lacks balance? When your microbiome becomes unbalanced, you may become unhealthy and even susceptible to chronic disease, many different kinds of chronic disease, including cancer. But with the help of your superhero, your microbiome always strives to return to a natural, balanced state. Eating healthy food can help your superheroes to be more effective in keeping you healthy. Your microbiome plays a starving role in many processes from digesting food and affecting mood and energy levels to preventing invasions by harmful microorganisms. Your heroes are working hard to keep your gut healthy every minute of every day. We want you to understand that a healthy microbiome means a healthy you. Okay, after understanding the human microbiome, let's second discuss the role of microbiome in cancer. Dysbiosis means that you have an imbalance in the different types of microorganisms living in your body with loss of good bacteria and overgrowth of bad bacteria or pathogenic bacteria. Different bad bacteria can cause different kind of disease. From this picture, you can notice that some bacteria are associated with infection, some associated with inflammation, some are associated with immune disease, some are contributed to cancer. You see the balance in dysbiosis, this bacteria overgrowth, but good bacteria only one, but part of bacteria do nothing, just like a passenger. So some good bacteria getting less, bad bacteria getting more, that means dysbiosis. In this regard, therefore, the dysbiosis has been implicated in a wide range of disease, including inflammatory bowel disease, IBD, autoimmune disease, asthma, neurological disorders, metabolic syndrome like type 2 diabetes or type 1 diabetes. In particular, dysbiosis can induce cancer. Due to the particular location, the gut microbiome in colorectal cancer may play a very important role. Let's start the discussion for the correlation of microbiome with the initiation and progression of colorectal cancer. To examine the contribution of gut microbiome in colorectal cancer CRC, we compare the gut microbial community difference in colorectal cancer patients as compared with the healthy subjects. Is the same or different, and based on the stool in terms of microbiome. We evaluate this microbial composition by using a shotgun microbiome shotgun metagenome sequencing. What is shotgun metagenome sequencing? It is a technology and is a second-generation sequencing and used to sequence the genomes of the entire microbial community in the gut, and without the need of culture of them first, so we can capture all the genome and produce from the gut microbiome. What look like the CRC patient gut microbiome compared with the healthy subjects. And this slide shows that you can see here the red color is colorectal cancer patients, which showed a very lower microbial diversity, suggesting an unbalanced and healthy microbiome composition as compared with healthy control subjects. In particular, we identified the particularly enriched potential pathogenic bacteria in colorectal cancer patients, including the first one, Streptococcus and Neuroblast. And this bacteria is significantly enriched in colorectal cancer. The function is totally unknown in terms of the correlation with cancer. In particular, some enriched bacteria in colorectal cancer patients are known oral pathogen, which showed a strong positive association, indicating they may work together as a team to promote the initiation and the progression of CRC, colorectal cancer. And based on this observation, we want to see whether the change of gut microbiome in colorectal cancer patients as compared with the control subjects, is this a cause or consequence, a chicken or egg? And with this question, we do the series analysis. And before that, I'll give you some background information. Colorectal cancer developed through a multi-step process. It's not suddenly occur. It's for many, many years, which involving a benign adenoma or polyps to cancer. So adenoma or polyps is benign, and this progression can take years or even decades. And then to understand the microbiome contribution is not only a consequence, perhaps it's also a trigger factor or early event. So we studied, according to disease progression, we did a biopsy from normal adenoma polyps and colorectal cancer. And then from these slides, you can see the microbial changes according to the disease progression from normal adenoma to colorectal cancer. And there is a shift and gradually increased. Of course, some bacteria is declining according to disease progression, indicating the cause-effect gut dysbiosis in the development of colorectal cancer. So it's an early event, but not a consequence. Then we ask if the microbiome changed because the dysbiosis in colorectal cancer, we identify whether it can promote colorectal cancer progression. With this question, we performed additional investigation. We collect the stools from colorectal cancer patients. We collect the stool from healthy subjects, or put them together, and then we gavaged. It's also called fecal microbiome transplantation. We gavage the stool from CRC patients and healthy subjects randomly to the germ-free mice. Okay, and I give you some explanation for the germ-free mice. And the germ-free mice, and according to the name, is like all microorganisms are housed in a tightly controlled and monitored isolator. You can see this is the isolator to prevent contamination. And with these germ-free mice, we only gavage the stool from CRC patients and healthy subjects to see what's happened, and based on this mouse model. The stool, we found that the stool from CRC patients was demonstrated to promote colorectal cancer formation in mice. These findings provide the first evidence that the stool from colorectal cancer patients induce colorectal cancer initiation and the progression. It's very bad, and due to the dysbiosis, due to a lot of overgrowth of bad bacteria in the gut in the CRC patients, not only early event, but can also contribute to the disease progression of colorectal cancer initiation and progression. All the stool bacteria play an important role, and then it is important to identify and characterize novel driver bacteria, the super guy that associated with the pathogenesis of colorectal cancer. This is the enriched bacteria identified in colorectal cancer patients as compared subjects. These bacteria, the first four, are new one, no reported from others, and whether the bacteria can contribute to the cancer formation. So we choose the first one, Peptrospectrococcus anaerobus, to see whether it's a novel candida bacteria, and then to contribute to CRC formation. And then we isolated these bacteria from the stool. We want to see these bacteria, whether it's a causative role and the underlying mechanism of these bacteria promoting colorectal cancer formation, and based on interplay with the host cell. Before this bacteria study, we want to see whether these bacteria have a closely interplay and with the host colon epithelial cell. This picture and shows under electron microscope, we observed that T anaerobus can attach and invaded into the colon cell. So with this observation, and then we did a very, very comprehensive investigation from mouse model, from cell line model, and from mechanistic investigation. We confirmed that T anaerobus is a new pathogen in promoting the initiation and progression of colorectal cancer, and through a direct interplay with the colon epithelial cell by activating oncogenic signaling pathway by suppressing anti-tumor immunomicroenvironment. So this is an example for a driver bacteria in contributing to the initiation and progression in colorectal cancer. And then we move on to stomach cancer because it's also closely correlated with the gut microbiome. And then in the stomach cancer, we also did the genome sequencing in gastric cancer GC as compared to the superficial gastritis as a control. We identified significantly enriched bacteria you can see in the red circle. And in particular, one bacteria named Streptococcus aeruginosus is the top enriched bacteria in gastric cancer as compared with healthy subjects. On other side, we want to see H. pylori is the first carcinogen for gastric cancer according to WHO. And then we want to see, in addition to H. pylori, is there any bacteria also contribute to gastric cancer initiation and progression? To answer this question, we want to remove H. pylori to see what's happened, whether other bacteria associated with the disease progression, especially the precancer lesson to cancer formation. So we collect the samples. This is a large-scale population-based study. We collect the samples in a high-risk area in Shandong, Muping. We retrieve this sample, analyze the gastric biopsy samples comprising 102 pairs and before and after 1 year H. pylori eradication, and 100 pairs. You can see here 100 pairs before and after 1 year placebo. We did a microbiome sequencing to see what's happened with or without H. pylori infection. After H. pylori eradication, we identify some bacteria that are positively associated, especially the red column, is correlated with persistent progression of chronic gastritis and precancerous lesions. And this atrophy intestinal metaplasia is the precancerous lesions. Without H. pylori, still the disease is still progression, persistent progression. So this bacteria you can see the red row, and then is a newly identified bacteria in additional H. pylori may contribute to the gastric cancer formation. Interestingly, we overlapped with the enriched bacterial gastric cancer. This overlapped bacteria, we highlight Streptococcus and this aeruginosus and this particular bacteria to further study. So, and this Streptococcus aeruginosus is oral pathogen. The function link and between this bacteria with cancer was not reported, but is highly enriched in gastric cancer, but also associated persistent progression, chronic gastritis, and also precancerous lesion. And based on the human study, we want to confirm this observation, and this only we can do by using different mouse model. We infect the mouse in germ-free or conventional mouse, and this bacteria, Streptococcus aeruginosus, induce acute gastritis after 2 weeks infection. So H. pylori was chosen as a positive control. Both can induce acute inflammation after 2 weeks. After long-term infection, as aeruginosus induce chronic gastritis, atrophy, metaplasia, and then even dysplasia. Atrophy, metaplasia, dysplasia is a precancerous lesion. It's before gastric cancer, after long-term infection, even germ-free by only this bacteria can also induce such kind of precancerous lesion. The function in gastric cancer also studied as aeruginosus can promote gastric cancer in different mouse model. In this regard, we give a conclusion that a novel bacteria named as aeruginosus can induce chronic gastritis, precancerous lesion, and also contribute to gastric cancer initiation and the progression. So it's promote gastric cancer. We did a mechanistic investigation for the interplay of this bacteria with the gastric cancer initiation and progression. And this work also already published last year in Cell. So, and based on this representative bacteria in colorectal cancer, representative bacteria in gastric cancer, and then also the important role of the gut microbiome in health and disease, I would like to summarize that. Keep your microbial complexity and stability is good for your health. Thank you so much for your attention. Thank you.